3. Cell cycle mitosis. Most essential learning competencies are characterize the phases of the cell cycle and their control points. Describe the stages of mitosis given the diploid cell where 2n is equals to 6. Explain the significance or applications of mitosis and identify disorders and diseases that result from the malfunction of the cell during the cell cycle. For your learning objectives, you're going to describe mitosis, explain the phases of mitosis cell cycle, identify and describe the stages of mitosis, understand the significance or applications of mitosis, and identify disorders and diseases that result from the malfunctions of the cell during the cell cycle. Here are some essential questions related to the topic. Have your body changed in the past 6 months? I mean, how tall are you? Or how much do you weigh? Are you taller? I mean, how do you know you're growing taller? Have you experienced a broken bone recently? What about bone? How a wound heals itself? I mean, how does your body repair itself? Have you also noticed how a cut on your finger is gradually repaired in a matter of few days? This is enabled by a process called mitosis. This process is responsible for regeneration and repair. It helps in cell growth and development. Cells can grow old and wear off, or they can get bruised and injured, but eventually they repair and regenerate. What is mitosis? It is the process that ensures that the daughter cells produced have identical sets of chromosomes. It means that each new cell receives one copy of every chromosome that was present in the original cell. It produces 2 new cells that are both genetically identical to the original cell. What kind of cells will perform mitosis? All body cells class except the cells. Where does mitosis occur in plants and animals? It takes place in the development of an embryo in animals and plants, followed by fetal development in animals. The single cell produced by gamete fusion divides by mitosis, forming entire new organisms. The rapid growth of the radicle and plumule in the germinating seed depends on rapid mitosis. In animals, mitosis for growth takes place throughout the organism until the animal is an adult and growth stops. In plants, mitosis takes place throughout life in the growing regions called the meristems. The cells of the skin and bone marrow are sites of active mitosis, replacing skin cells and red blood cells that only have a limited life. When an area of tissue is damaged, internally or externally, mitosis is used to repair the damage. Mitosis is a part of the cell cycle. Mitosis is a part of the cell cycle, and it is preceded by the S phase of interphase, and usually followed or accompanied by cytokinesis. Replication of chromosomes and synthesis of proteins required for spindle fiber formation are formed prior to the beginning of mitosis. Mitosis is divided into the following phases based on the completion of one set of activities and the onset of the other. Interphase. It is a part of the cell cycle where the cell copies its DNA as preparation for the mitotic phase, and it is often termed the most reproductive phase of the cell cycle. A series of metabolic changes occur during this phase, all which are divided into three subgroups. We have the G1 phase, GAP1, or the first growth phase. It is also called the free DNA synthesis phase. It is the longest phase of the cell cycle, and it is followed by the mitotic phase of the previous cell cycle. It is also termed as the resting phase, as no DNA synthesis takes place during this phase. However, during the G1 phase, several cell organelles increase in size and cell rapidly synthesizes different types of RNA and proteins. S phase, or also called as DNA synthesis phase. S phase involves the replications of nuclear DNA and the synthesis of histone proteins. The replication of cytoplasmic DNA can take place at any phase in the cell cycle. Thus, at the end of the S phase, each chromosome has 2 DNA molecules and a duplicate set of genes. This phase lasts for about 6 to 10 hours. Then next we have the G2 phase, or the post-DNA synthesis phase. G2 phase is termed the second gap phase or resting phase of the 8th phase. During this phase, the synthesis of RNA and proteins required for the cell continues. By the end of this phase, the cell enters the division of the mitotic phase of the cell cycle. Prophase. It is the first stage of mitosis, which is characterized by the appearance of thin thread-like condensing chromosomes. During prophase, the cell becomes spheroid, while the cytoplasm becomes more retractile and viscous and thin. As prophase progresses, the chromatids become shorter and thicker, and two sister chromatids of each chromosomes are held together by a special DNA-containing region called the centromere. Lastly, during prophase, the nucleolus gradually disintegrates, and this marks the end of prophase. Prometaphase. It is initiated with the breakdown of the nuclear envelope, which enables the interaction of spindle fibers with the chromosomes. At this stage, the chromosomes are violently rotated and oscillated back and forth between the spindle folds because their centromeres are capturing the ends of microtubules and are being pulled by the captured microtubules. By the end of this phase, the sister chromatids are attached to the spindle fibers on the opposite end and are held on the metaphase plate. Metaphase. During metaphase, the chromosomes are shortest and thickest. The centromeres of the sister chromatids occupy the plane of the equator, forming a metaphase plate, and the arms remain directed towards the poles. Anaphase begins abruptly with the synchronous splitting of each chromosome into its sister chromatids, called daughter chromosomes, separating for the centromere. There is a movement of chromatids towards the pole due to the shortening of the microtubules. During their poleward migration, the centromeres remain forward so that the chromosomes appear like letter U, B, or J shape. The end of the migration of the daughter chromosomes to the poles marks the beginning of the telophase. During telophase, the events of prophase occur in reverse sequence. Events like the disappearance of mitotic apparatus, reductions in the viscosity of cytoplasm, followed by synthesis of RNA takes place during telophase. The chromosomes resume their long, slender, extended form, and the nucleolus reappears at the end of telophase. Cytogenesis is of cytoplasm, which is followed by mitosis, resulting in the formation of two separate daughter cells. It usually begins in anaphase and continues through telophase and into interphase. In animals, cytokinesis occurs through constriction and chloroformation. In plants, cytokinesis occurs by cell plate formation, as constriction is not possible due to the presence of a rigid cell wall. And this is the summary of the stages of cell cycle mitosis. Mitosis constitutes a comparatively small portion of a complete cell cycle, but it is one of the imperative parts of the cell cycle. German physician and cell biologist Walter Fleming coined the term mitosis in the year 1882. He explained the process of how cells split and separate their chromosomes. He first discovered mitosis in year 1870 and reported this discovery of chromosomes in year 1882. Based on the given illustration, the metaphase in a salamander cell was drawn by Walter Fleming from a stained slide. Mitosis plays a crucial role in a living organism's life cycle. However, the level of significance may vary depending on the type of organisms, either multicellular or single-celled organisms. In a unicellular organism such as bacteria, mitosis helps in asexual reproduction as it produces an identical copy of the parent cell. Another example of the eukaryotic unicellular organism is amoeba. An amoeba uses cell division for the production of new individuals. In the case of multicellular organisms, mitosis helps in growth and repair by producing more numbers of identical cells. For example, plants and animals depend on cell division for their growth by addition of new cells. It is also used for repairing the injured tissues or replacing the worn-out tissue by regenerating new cells. To better understand the cell cycle mitosis class, let us have a sample analogy, the cell checkpoints or stoplight. There are times when the cell is supposed to stop and check itself. These are disabled in cancer cells. As you can see in our illustration, between any two stages, there are stoplights where the cell checks the previous step completed correctly and the cell is not damaged. If the cell doesn't pass the stoplight, it dies. As we recall class, mitosis makes two new cells from the parent cell. As cells grow, they go through a number of stages. First, the cell grows normally in the blue region, or decides not to grow, and sits outside the circle for a time. Then the cell duplicates all its DNA in the red region. In the next phase, the cell grows just a little more in the green region. Finally, the cell divides into two new cells in the purple region, a process called mitosis.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
The term cloning describes a number of different processes that can be used to produce genetically identical copies of a biological entity. Researchers have cloned a wide range of biological materials, including genes, cells, tissues, and even entire organisms such as sheep. In reproductive cloning, researchers remove a mature somatic cell, such as a skin cell, from an animal that they wish to copy. They then transfer the DNA of the donor animal somatic cell into an egg cell or oocyte that has had its own DNA containing nucleus removed, and this young animal is referred to as cloning. Molecular cloning transfers one gene from the cell of one organism to the cell of another. Tissue culture. It is a method of biological research in which fragments of tissues from an animal or plant are transferred to an artificial environment in which they can continue to survive and function. The cultured tissue may consist of a single cell, a population of cells, or a whole or part of an organ. Cells in culture may multiply, change size, form, or function, exhibit specialized activity. Muscle cells, for example, may contract or interact with other cells. Stem cells are undifferentiated or blank cells. This means they are capable of developing into cells that serve numerous functions in different parts of the body. Most cells in the body are differentiated cells. These cells can only serve a specific purpose in a particular organ. For example, red blood cells are specifically designed to carry oxygen through the blood. Stem cells are cells that haven't differentiated yet. They have the ability to divide and make an indefinite number of copies of themselves. Other cells in the body can only replicate a limited number of times before they begin to break down. When a stem cell divides, it can either remain a stem cell or turn into a differentiated cell, such as a muscle cell or a red blood cell. Since stem cells have the ability to turn into various other types of cells, scientists believe that they can be useful for treating and understanding diseases. Researchers hope stem cells will one day be effective in the treatment of many medical conditions and diseases. Stem cell therapies may offer the potential to treat diseases or conditions for which few treatments exist, sometimes called the body's master cells. Stem cells are the cells that develop into blood, brain, bones, and all of the body's organs. They have the potential to repair, restore, replace, and regenerate cells, and could possibly be used to treat many medical conditions and diseases. Today, doctors routinely use stem cells that come from bone marrow or blood in transplant procedures to treat patients with cancer and disorders of the blood and immune system. Mitosis is closely controlled by the genes inside every cell. Sometimes this control can go wrong. If that happens in just a single cell, it can replicate itself to make new cells that are also out of control, and these are cancer cells. This picture of cancer cells show that cancerous cells lose the ability to stop dividing when they contact similar cells. Cancer cells no longer have the normal checks and balances in place that control and limit cell division. The process of cell division, whether it is normal or cancerous cells, it may also go through the cell cycle. Cell mutations within an individual aren't always uniform. Some cells may have a mutant version of a gene, while others have the normal version of the same gene. Geneticists refer to this condition as mosaicism. In somatic cells or cells other than egg or sperm cells, the individual may not be affected by the mutations. But if the mutant genotype is widespread and harmful enough, the mutation can have a major impact. The term mosaicism is used to describe the presence of more than one type of cell in a person. For example, a person may have some of the cells in their body with 46 chromosomes, while other cells in their body have 47 chromosomes. And an example of mosaicism is the mosaic Down syndrome. Chromosomal abnormalities. So we have a certain structural abnormalities. A chromosome structure can be altered in several ways. We have deletions, a chromosome which is missing or deleted, duplications, resulting in extra genetic material, translocation, a chromosome which is transferred to another chromosome. So during pregnancy, an error in mitosis can occur just like the error in cell division in meiosis. If the chromosomes do not split into equal halves, the new cells can have an extra chromosome or have a missing chromosome. This is another way a baby can be born with a chromosome abnormality. Effects on organelles. Key organelles that make mitosis go as planned include the nucleus, mitotic spindle, and microtubules. As we previously discussed, the analogy about checkpoints and spotlights of mitosis, checkpoints during the cell cycle help correct errors and prevent serious problems such as chromosome abnormalities. And when it comes to cell division, cancer cells break just about all the rules. Cancer cells can divide without appropriate external signals. They do not exhibit contact inhibition. It means cancer cells no longer respond to this stop signal. It can divide without receiving that all clear signal. These mutant cells are even more abnormal than the parent cell. In this manner, cancer cells can evolve to become progressively more abnormal. Continued cell division leads to the formation of tumors. So the process of mitosis is significant in both cell division as well as cell reproduction. Continuous mitosis results in the increase in the number of cells, enabling the organism to grow from a single cell to a complex living organism. Different cells in the body, like the cells on the skin and red blood cells, are continuously replaced by mitosis. Also, it involves in the repairment and regeneration of body structures.
Have you ever been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together. Working together as part of a body tissue. Body tissues working together as part of an organ. Organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle, and that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size, but let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you are bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows that splits the cytoplasm allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated. They are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there's a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So, what is the cell cycle? The cell cycle is often represented as a pie chart like this. Cells are either in one of two different phases. A phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells, but cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation, for example, you do not want it to divide, because then it will create another cell that has this same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide this cell cycle pie chart. We have G1, S, G2. All three of those are part of interphase. Then we have M phase, where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. Then G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase, where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay then, moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, the chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions, and there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle, and the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2, N, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not an O, because if it was an O, then it's a go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to end phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate. A topic that definitely continues to be researched.
Activity sheet number 1. The diagram below shows 6 cells in various phases of the cell cycle. Note, the cells are not arranged in the order in which mitosis occurs. Use the diagram to answer the following guide questions. 1. How can you tell which diagrams are the end of the cell division? 2. What are structures seen inside the nucleus? 3. Are the cells depicted plant or animal cells? Explain your answer. 4. Use the diagram at the bottom of your answer sheet. Draw the phases or stages of the cell cycle and arrange them in correct sequence. 5. Why is mitosis important? Give at least 2 reasons.